Well, good day, and welcome to our webinar this morning on eggshell quality. Welcome also to our Spanish-speaking customers. Bienvenido también a todos nuestros clientes quien hablan español. La próxima semana tendremos la misma clase. Si desea verlo el próximo martes en lugar de hoy, puede visitar www.chickmaster.com y registrarse ahí ahora. My name is Mark Sintel, and I'm the Vice President of Customer Care for Chickmaster, and I just would like to welcome you. Really delighted that you joined us today. While we can't see each other in person, we can connect this way, and I'm really glad that you would choose to spend an hour or so talking about eggshell quality issues in your hatcheries. This is not necessarily our area of expertise as a company, but it is an important topic for your hatchery and for your farms. And our motto is that your hatchery is our priority. And so we thought this would be a topic that we could facilitate a discussion on. And so if you have any questions during the presentation, please use the Q&A button that's at the bottom of your screen. So press Q&A and enter your question, and we'll address those throughout the presentation. At the end, you could also raise your hand if you'd like to ask your question live, and we will unmute your microphone for a moment at that time. So please be aware, though, that we are recording this presentation, and so we can make it available to others. So you may prefer to type your question instead of being recorded. Our presenter today is Mohamed Ait Lachgar, and Mohamed has worked in the incubation industry for like 20 years. And he recently rejoined Chickmaster, rejoined our team after spending a few years elsewhere in the industry. And we are so delighted to have him back with us as one of our senior customer service leaders. Mohamed is not an embryologist, but he has worked in engineering and worldwide installation support and customer service, project management, and he's picked up so much over the years that we thought he would be a good person to present and summarize some of the things that we've learned about eggshell quality. We also have Dr. Carolina Diaz with us. Carolina is an embryologist and a hatchery specialist, and she's going to be answering some of the questions that you send in and helping Mohamed if we need to reach for her expertise. So without any further introduction, welcome again, and I give you Mohamed.
Hi, and good day to everyone then. Thanks again for joining us today for that webinar, in which we will talk about various ways for you to evaluate the egg quality. Why do you need such information, you may say? Well, I would like to share with you a larger understanding on all the relation between the egg production, its structure, and the different ways to evaluate them, combined also with some scientific results, hoping that will give you criteria which positively gets impact on your final results in the production on your hatchery. To reach the goal to give you the necessary information, we will go through these 6 steps shown here. They are made in a such way to give you all the bases on some theoretical principle aspect, in order to know why and how evaluate the eggs quality. Also, I want to tell you that I come from technical background, and with the years visiting hatchery with embryology experts, I found that knowing the basis of the egg's biology is really helping to understand the relation between the functionality of the incubation equipment and also to get them fine-tuned correctly for the production benefit. Let's start with the introduction now. Which came first, the egg or the chicken? This is a scientific question on the evolution of nature in the reproduction process. But the important aspect for us in that question is the quality relation between both sides is critical. Indeed, the good quality of the eggs has a high good potentiality to give healthy, well-developed chicks. And a healthy hen chicken gives a good egg quality for the healthy embryo development, and that keeps going in circle. If something breaks in that quality circle, then problems start in both sides, farms and hatcheries. Interrelation is important to understand here. Your goal in hatcheries is to produce chicks using eggs that you get from someone else, right? So the daily hatchery work is to copy nature. Your main goal is to manage and get all the best conditions for these eggs that you receive to become a healthy, alive bird. Looking from outside, it can seem magic at first, but you know by experience that the good results come from a combination of knowledge in the hatchery on technical equipment and some biological basis. This in order to be able to track good practice and understand the cause of the issues when they appear, to keep the production in highest level with the best quality and the best hatchability. Compared to the natural process of bird laying and embryos development, your challenge is to produce using a high volume of eggs. The best conditions should be given to the eggs for the best embryo development, which start at the very beginning, like the following video shows, with the cells development starting at day 1. But the egg quality starts even before that day, as we will see. All these necessary technical and ambiental conditions for the embryo development were explained by my colleagues on previous webinar, and I recommend you to get in Chickmaster website to take a look at them. Egg being the main start product in the hatchery process, its quality is essential, and we will focus in this presentation on some of the points which can help you to identify the egg qualities you are receiving. Step 2, let's see together the process and the structure of an eggshell to understand why it is so important in relation to the egg quality and in final to the embryo development. Just to precise here that even if some point can refer to various types of hatching bird, I will focus and concentrate on the next slides in the hen chicken eggs. The eggs production system is named oviduct, which is a tube-like organ laying. In a mature hen, it is approximately about 25 to 27 inches long. This oviduct is divided in 5 parts and has its own functions to the egg production. The egg formation starts in the ovarian follicle, in which there is already all the ovule. Each day, an ovule leaves the follicles and reaching the infundibulum where it is fertilized. This takes approximately 10 to 15 minutes in the process. Then, the fertilized eggs goes into the magnum during 2-3 hours, where the ovule, the yolk, gets surrounded by the albumin, the white, which is normally opalescent color instead of white. Transiting into the isthmus during 1-2 hours, the yolk plus albumin gets the internal and external thin shell membranes. And now, this is here in the uterus that takes place the 20 hours long process of hard eggshell formation. So we can see here already the direct relation between the hen health condition and its impact on the eggshell quality, right? As this is the longest time process in the hen. Finally, the eggs is laid via the vagina in few minutes. So the egg is laid now. How all this 24 hours process we just seen results inside the egg structure itself? Well, we have in the fertile eggs the panda nucleolus and the blastodisc showing the embryo development starting point. The chalazae are 2 spiral bands of tissues that suspend the yolk in the center of the albumin. The function of the chalazae is to hold the yolk in place and explain why the yolk will never move from the middle of the eggs. The yolk is structured with various separated parts. And also we have various layers of the albumin, which is finalized by the eggshell membrane itself. So for you to know a little bit of this structure can help during the egg breakout, for example, to determine possible issues like the quality of the albumin, the inner membrane quality, etc. But that is a total topic for another webinar. As the eggshell production is the longer process, as we've seen, we will focus on it as a support to get direct information on the egg quality. Just a remark, The air cell is a result of the contraction of the albumin under a thermomechanical effect. Indeed, this contraction is caused by the difference of temperature between the inside hen body temp, which is at 102 Fahrenheit, which the egg also has at the laying time, and the outside ambient air where the egg is laid in the farm, around 65 to 85 Fahrenheit. So the liquid albumin contracts more than the shell, and the inner membrane separates then from the outer membrane to form the air cell. So the air cell is formed just a few minutes after the egg was laid during the cooling period times. As we saw the structure of the eggs, the repartition of the different components are as follows. The total weight of the eggs is divided in three parts: the yolk, the albumin, and the shell. The shell itself is the lowest in percentage of the weight, but it is the longest one to produce. In the composition distribution, there's mainly water, and the shell represents the majority of the 11% of the mineral distribution in the composition of the eggs. So the embryo is finally surrounded and protected by 4 levels of layers shell membrane, as you can see on the top right of the picture. The dry shell is composed with the one main mineral, the calcium, and few others. You can understand now that to maintain this high level of calcium for each egg the hen is producing during his life, it has to be provided to the hen by the food. And its concentration is important, and we will see why in a moment. On the previous picture, we saw just a simple hard eggshell. But if we look very closer, we can see that it is made also with many layers. It is not just one block. In the production process, it starts with small drop of calcium, as shown the scanning picture. And then the calcium structure raises up during 20 hours, as you remember, to become more a block-like at the end of the process. One very important part of the embryo development is on the chorioallantoic membrane, also named chorioallantois. Why? Because that chorioallantoic membrane allows the exchange of gas, such as oxygen, and it also plays an essential role in bone formation in the embryo by transporting the calcium into the embryo from the eggshell. The reason why that membrane is so close to the eggshell structure. All the transfer from the outside ambient condition and the embryo inside the eggs, means inside the eggshell, are clearly depending on the development inside the end of each of the following layers. If one of them is not properly produced, it will directly impact the embryo development. Indeed, if one layer is too large or others are too thin during the process, it will not support the different needed conditions during the incubation process for the embryo to develop well. As you see, there is layers on top of layers, and all are interdependent from the very bottom start. We don't forget the cuticle, which is the ultimate separation between the eggs and the outside, which acts first to help laying the eggs, and then to protect it from the outside condition. And also, it helps for some regulation in gas and moisture during the incubation, for instance. We often see the eggs setters, which are usually coming from the floor eggs that were washed in the farm, and during this washing, they have damaged that bioprotective cuticle. So it is very important point to check if that cuticle is clean and has its complete integrity. It is clear now that the eggshell quality is therefore one of the most important checks you have to do, as it will directly impact the condition of the embryo and its development if he cannot regulate one of the many functions that he has to do. They are all related to the hatchery process management, which were explained in the previous webinar, like the preventive maintenance, so the machine can support best and maintain the embryo temperature all along the incubation process. It's also important to know the hatchery ambient air and ventilation, which helps to support the eggs moisture lost and the gas exchange. So the eggshell structure and its good quality at the start will allow all this regulation to be supported according to the embryo needs. So now that we get the main reason of the importance of the eggshell quality, which support the embryo development, and so the impact in your chick production, what are the methods to measure its quality? Well, it is obvious that the main aspects are visible by every first look, like the shape of the eggs, its size, and its color. But there is more possibility to control it, and which helps science and experts to get better understanding of its function and quality in relation to the chick's quality. Here are the list of the most used methods that we will describe you. And if you know any other, just let us know in the chat. So the main one are the specific gravity, the shell breaking strength, the percentage shell, the shell thickness, and the shell color. Let's start with the specific gravity. From the science, physical science, it's a non-invasive method to control the thickness of the hard shell. The method itself uses a precise salt concentration, which is made using some tools. When that solution is prepared, it is just a matter to put the sample of eggs to control in that solution, and the eggs that float has a specific gravity below the normal value, which was defined by research as a 1.08 specific gravity. The specific gravity value of an egg indicates the quantity of shell relative to other components of the eggs itself. These values usually decline as the hen ages. This is partly due to the size of the eggs increasing more rapidly than the shell itself during the process. Using this specific gravity in a sample of eggs from same flock can give you already information if these eggs are good shell quality that will not easily break during egg processing, for instance, like the egg grading, putting eggs in the setter trays during the transfer. It is a good tracking indicator. So the second point to control the eggshell is the breaking strength. It's a mechanical test in the eggshell resistance. It's normally uses tools to put a constant pressure in the eggs until it breaks. This can help to define if the egg from a certain flock has a good homogeneity and eggshell resistance, and we will show some value on the last part of the presentation with scientific parameters. Here, you can see some data example of measuring from Lohmann. We can see here that in their test, the homogeneity is different between the two flocks, the line A and line B. Also, it's good to know that in some egg grading machine in the industry, it proposes what is named a crack detector. This doesn't break the eggs, but use an electronic to define the sound feedback of the eggshell under a vibration hammering. So when the eggs come inside the hatchery with this process, they can already remove the eggs which has a very thin crack that can not be seen by eyes. The other point is the shell color. It provides some feedback on the hen feeding composition, and in final, a very interesting relation between the percentage hatchability and the darkness of the shell. Be patient, I will show you some data at the end of the presentation in the scientific results slides. The other points to measure the eggshell quality is the percentage of shell. It's another way to get the data of its quality. It's easy to do by weighting the eggshell alone compared to the weight of a full egg. And for example, this was used by this research shows below, and it clearly shows that the percentage of shell is decreasing as the breeding flock ages. The shell thickness in research is stated as a major indirect parameter. It can be measured on clean broken shell with a digital tool, or also in a fertile eggs using ultrasonic instrument like shows. With this control, you can know exactly what is the thickness of the eggshell compared to the edge of the flock, and that will give you a good indicator if the flock and the eggs are in the good quality. Now, there is another point you can do to see the eggshell quality is using the visual aspect. So we will present you now the easiest way to do that. As you can see, there will be some eggs that you will not like to receive in your hatchery, right? But this visual check can give you some feedback on what is happening in the farm also. The main eggshell irregularities and defects were already been defined by the expert and genetic breeders, and shows in this type of posters as you may already have in your hatchery. The most common you will see is dirty, for example, or blood-stained eggs, which is caused by overweight pulley, or with very young, which can also be related to poor hygiene or mechanical system in the laying farm. Until that is found and solved, the problem can keep going same in a long period of time in the farm. Each type of defect has a cause in the farm, and this list should give you a red flag when you have too many of these visual defect eggs in a batch. For example, in season changes, the heat stress can cause cracks and corrupted eggs. So, using this table, when you can identify yourself by the eggshell defect aspect, the problem can help you also to relate your own hatchability result to this egg quality. As an example of my experience, when I visited a couple of years ago a hatchery where we seen many dirty and blood eggs and some with misshaped form, we already knew that the hatchability was impacted. And then the hatchery manager confirmed and dropped following changes of egg farm suppliers. He did a control of the following egg batch and he removed all these defected eggs, after which he contacted the farm manager explaining him the situation. And together they solved the problem, and the hatchability was getting up after that because he was receiving a better quality eggs. With the previous visual defect details list, you get many information on the factors impacting the eggs quality. What are the main ones? At the really beginning of the eggs process, the main factors are obviously the genetics. From each type, you will have the evolution of the egg weight, and for each week of flock age, and that evolution impact the eggshell quality. And in support of the genetic, the food quality and quantity will provide or not the necessary element for the hen to produce a good egg. The third point is the microbiological environment of the breeders, as if they need a lot of disinfection or medicine that will finally get into the eggs and impact also the eggs process quality. Then, what's happened after hen in a good health has given many really good eggs quality. There is still a lot of process, many steps, which can impact the eggs before it gets into the hatcheries. Here are the main ones, and any of these steps can impact the egg quality. If the eggs are really good and strong at laying, still, each one of these step process need a really good quality management, because a good eggs laid in the hen farm can be contaminated or get cracked during the egg collection, or be damaged by the bad disinfection process, or during the transportation from the storage to the hatchery. After all this process, if they are well respected in quality management, the eggs finally arrive in your hatchery with the best potential capacity. So remember, any one of these processes can damage the original egg quality and lower your potential of good hatchability. And it's good for you to be aware on all of these steps in your own site.
Mohamed,
yes.
Let me, before you move forward, let me just back up just for a moment. We do have a question that someone put in the Q&A, and that was about, is there any negative effects of urates or blood on the shell? Anything that you could speak to? And actually, I just saw Carolina answer this question saying that both are indicators of breeder health issues. The blood will be an indicator of the reproductive system of the egg having been affected. Blood should not be accepted as it can contaminate eggs. Sometimes it can be caused by a microorganism action or by rough hen handling. So do you have anything else to add to that on negative effects of urates or blood on the shell beyond what Carolina has answered?
Any dirt that you can see in the eggs will have a negative impact during the incubation, because this is a potential growth of infection, because when you have dirty and blood on the eggs, you have some cells and bacteria that can grow, and indeed during the 18 incubation process that can be grow inside the setters and contaminate the other eggs. So if you have few of them, it can relate that the eggs, a couple of hen can have problems, but if you see too many in a batch of eggs that you receive, then it can relate to a most critical and important health issue in the farms, and that can really impact negatively and have a contamination inside the hatchery. So like Carolina said, it's good to see the percentage of them, and if it's too many, to get back to the farm and to try to avoid to incubate them.
Okay, very good. And one more question. We see in the picture you have here that the disinfection stage there is being done wet. Is that a suggested method or preferred method of disinfection?
No, it's really to show that this process can also impact. It's not a recommended one. We used to provide more the disinfection by gas, but it was also a picture here to show and to impact visually that it's impacting. But we don't recommend a wet disinfection.
Okay, thank you. And so, one more question, sir, on this, and Carolina may be better to answer this. Just after the egg is laid, the pores of the egg take how long to close during the cooling period to normal room temperature? When would it be best to disinfect freshly laid eggs?
Well, it takes a few minutes, so that's the reason why as soon as you can get the eggs to, as soon as you can collect them more frequently will be better. Just because you want the disinfection to get inside the egg, just in case it gets contaminated.
So just a few minutes to close is what you're saying?
A few minutes, yeah, right.
Okay, very good. Thank you.
So in all the process we just seen before, the temperature should be controlled perfectly to avoid thermal shock. Here, there is a chart shows the evolution of the temperature, and we should drop down from the farm to the egg store, and then the raise up before the incubation to avoid any up and down or backflow of the temperature level. And in some of these steps, the disinfection has to be defined to avoid too many times and too high concentration of disinfection in the eggs before they get in your incubators. So here, the most important you have to see on this graph is when the egg is laid is already between 102 and 106 Fahrenheit. And during the time, you have to drop down this temperature up to the egg store, so then you can keep these eggs in your egg store for the long period of times. If during this drop down, the temperature goes down and up again, you can have problem of cells starting to develop and the embryo starting to be developed before it gets to the egg stores. And then it's reduced the potentiality of development of the embryo. And also when you get them from the egg store, depending the temperature, if it's very cold room or a standard room at 80 degrees Celsius, for instance, you can either go by steps by a preheating, if you go to a multi-stage incubation, or directly to the incubator if you do a single stage. So like I said, you have to take a lot of precaution on the correct dilution and doses for disinfection. Because like Carolina just said, the disinfection will go inside the eggs. And if the disinfection dose is too high, it can impact the embryo inside the eggs itself. So now the eggs arrive in your storage in the hatchery. So the main aspect, as you know, to respect there is the uniformity and the conditions, the cleanliness of the egg store, and also a good thing would be to organize the egg store as referred blocks, the farm quality, etc., to avoid cross-contamination. Now that we've seen different aspects of the eggshell quality, let's go now for some scientific values which showing that all that we present today have a base from the field experience and they are reproducible. Sorry. I'm not expert and I will not go deep in this experience themselves, but I will share with you the understandable results. So for example, on the left graph here, it's result from a research done by Mr. Bramwell on the eggshell quality. It shows the relation between the specific gravity and the hatchability. You can see a drop when you have the specific gravity of 1.08, like the standard. The lower goes the specific gravity, it impacts the hatchability. Here you can see up to 20%. This is something you can do in your hatchery if you have a quality manager, for example, and start creating your own base and of reference of eggs quality control with your own breeders type that you are hatching. We saw before something how to relate with the darkness of the eggshell. So the right graph here shows a study that was done, and it shows that the lighter is the eggshell, you have a big drop of the hatchability. So it's a reference about the hen health feeding, which impact the eggshell color during the egg process production, and which has a direct impact on the hatchability, as you can see on these graphs. In this following research on the effect of the eggshell quality and hatchability, it was proved that the higher the specific gravity is, the less earlier mortality of the embryo we have. Looking of each experience from the each flock age, we see that the hatchability dropped down hard in a whole flock of 64 weeks. And also the mortality at 7 days in the farm from this flock is really going higher. In this other research, it shows the direct impact of the eggshell cracks of a 51 weeks old breeder age with a 4-day storage. We can see a very low hatchability on fertile eggs when you have a lot of air cracks. It goes up to 50% less hatchability. With the same previous flock as we've seen before, on this research, the final impact of the eggshell cracks type relates to the percentage of live body weight. We see a significant negative impact compared to the normal of the embryo development parameter. So this research shows also that the eggshell quality is critical for the embryo development, and any type of cracks will impact the embryo development, and sometimes you cannot see them. So you know now the multiple factors involved in the eggs production, how they impact the eggshell quality, how you can visualize them yourself, and also possibly make yourself some of the methodological control to create your own reference base and to improve your final result by knowing what level of eggs quality do you incubate in your hatchery. So in resume, by knowing this impact in the egg quality, you can also do your own egg quality process when you receive the eggs. So with all this information in final, which side do you prefer? There is always two ways. You can possibly keep taking and incubating all type of the eggs that you are receiving and just wait for surprise at the end of the results in the hatching. Or you can decide at the beginning to get the product that enter in the hatchery with the best quality and which respect all the steps from the laying place till your hatchery in the best condition. With that, you will be more confident that the chicks you will hatch will reflect the good management and practice of your hatchery place and teams, instead of depending on the eggs quality itself. So as the eggs are coming from the farms, and harmonious relation and communication can help prevent or stop quickly any deviation of eggs quality. And with today's presentation, we hope you have more knowledge to communicate between both sides. So in conclusion, we'll invite you to start making and keep in your side a good tracking record of your egg quality using one or more of the presented process and tools. Also, we invite you to start and develop a profitable communication and relation with your egg supplier, as you will find sometimes surprising cause of your lowering hatchability results. And for that, Carolina can have a good story of a visit she had in the hatchery. Carolina, you can tell your story.
Yeah, so a couple of weeks ago, we were with a customer and they were having issues with contamination. So they thought the fault was inside the hatchery. So they started using more disinfection process. They started doing some egg selection. I mean, being more strong with the egg selection. And by the time the process manager and the process manager realized that the purchase section of the company decreased the disinfection purchasing, and just because they changed some personnel in the farms and they weren't adding the correct dose to the disinfection and the nests and the eggs. So from that time, they correct the dose, they correct all the procedures, they got in touch with their uh, disinfectant supplier, and then they got some training on it. And from now, what they do is that every time the hatchery manager does a load, so he calls the breeder manager and he asks if he has seen any differences on um, eggs laid percentage, cool egg percentage, and if they have seen some of those kind of cases or any disease signal on the breeders, they will get a note just to correlate the farm information with the hatch, which is a good practice because they work as a team, and that way the hatchery manager is not struggling to know where is the main problem, so they can get a little bit of more control of the process.
Thank you, Carolina, for this example and real story. So we see that the hatchery is not an isolated place. It has to have a relation with the suppliers and the eggs breeders, so they can relate also some issue that can be experienced in the hatchery to the egg quality themselves. So for that, as a recommendation, we ask you to keep tracking a record of your egg quality receiving using our sample of quality assessment document, which will be available in our website. Set up and keep a good communication and information with your fertile egg supplier. And on last conclusion, you can also invite one of our chickmaster and embryologist experts during your meetings with the farms. With their expertise, they will certainly guide and help you to evaluate and set up in place the egg quality process by adapting it to your needs. Here is a non-exhaustive list of reference used in that presentation, into which you can learn more deeper aspects if you like this topic. As this presentation will be available in Chickmaster website, you will be able to get times to take this reference sample. Thanks a lot for having participated to that presentation up to this end, and I wish you have well appreciated its contents. The place is open for question and answer.
Right, Mohamed, thank you for pulling that together, and the end of that, where you recommend that for hatcheries who don't have a quality program in place, this is a good opportunity to start evaluating egg quality as it's coming into the hatchery, so you have a baseline to understand what's normal, and then to make sure you're having the conversations with the farms, because any number of things can affect the quality, and if you can identify them, then you can start to mitigate against what you're seeing. A few more questions have come in, and feel free to add others to our guests. We may not answer all of them live right now, but we will certainly get back to you with a specific answer if we didn't answer it live. And you are always welcome to email us at support@chickmaster.com or reach out to us any other way that you know how to, or you can find it at chickmaster.com, and one of our hatchery specialists will certainly call you back to talk about your specific concern. We have a few questions that came in about egg color. Do you know why darker eggs hatch better, why paler eggs hatch lower than darker eggs? Speak any more to that?
Yeah, mainly I don't have the deep look in the research itself, but I was reading some scientific search done by that, and it looks like the relation of if you have a specific breed hen, it has a specific color already as a norm, and the difference of the color of the eggshell is directly related to the hen health and also its feeding. So that's why in this research, they really made a clear view by the results that if the hen health and its feed is not perfect, it can directly impact the egg quality and directly the hatchability.
So are you saying that we're almost looking for variations from what is normal for that breed?
Yes,
okay.
Indeed.
So the variations may indicate there's a problem.
Yeah, if usually in the hatchery you receive a type of color eggs and you have a good result with them, on the next, or if you receive another batch and you see that the eggshell is whiter, you can already took note about this lighter color and refer that to the hatchability you will have. And with experience, you can make your own database to have a relation between the eggshell color and the hatchability. Okay. And refer that if you see that the hatchability drops too low because of this eggshell color difference, then it's time to communicate with the egg supplier to tell them that there is a problem here on the quality of the eggs they provide to the hatchery.
Okay. We have a question about cleaning eggs coming into the hatchery. What about dirty eggs? May we clean them by knife? Do you have any recommendations?
No, if you clean by knife, you will directly damage the cuticle. So it's not a recommended mechanical cleaning either than using water with detergent, etc. It's not a good way to clean up the eggs. So if you have a lot of floor eggs, what we usually recommend, it's concentrate all of them in one place and hatch them in one trolley or in one side of the machine. If you really need to hatch them. If not, try, if you have really dirty eggs, it's better to remove them to avoid contaminations.
Okay. We have several questions about standards. And so asking if there are standards for hairline cracks, dirty eggs, deformed eggs, white eggs, etc. And do we have recommended percentages for good and bad quality eggs? I'm not sure that we can give a general answer to that, but do you know of any reference material for standards for what is good for perhaps for different breeds? Or
no, there is no direct reference of the parameters, the value itself. It's more if you use a process, a control process like the shell thickness or the specific gravity, you have there in the control process a reference value. Like for instance, the gravity, specific gravity, it's 108. So many research define that this is the base value. So if you use this specific gravity of 0.8 and you do the check the eggs control with this system, and you have the value which are below or above, then it gives you a feedback of the quality itself.
okay.
And to do the eggshell thickness, for example, that all depend on the type of breeders, etc. It's maybe an information you can have from the eggs breeder farm or the genetic breeders directly. If not, you have to build your own database yourself by making measurement along the years to get you a good reference for your own productions.
Okay. We had a question about someone facing yolk infections at the hatchery. Any suggestions on how to go about solving this issue?
That might be more a question for Carolina, because I'm not that expert to go that deep inside the eggs.
Okay. Carolina, do you have any advice that you would give on how to address yolk infections at the hatchery?
Yes, first you should check it out if it is on a special flock or if it is on the whole egg bag you're receiving. Second, you should verify if you have increment on cool eggs, dirty cool eggs, or bloody cool eggs, and take them out. If you're not seeing those kind of things, it will be good that you can take some microbial samples and get them to the laboratory, so you can see if maybe there is something going on again on a special flock or if it is general. Also review your disinfection procedures and your doses.
Okay. Thank you, Carolina. I have a question about, again, about disinfection. During transfer, at transfer, if they perform wet disinfection again on the eggs, does it have any impact on hatchability? And if yes, what do they need to adjust in the hatcher settings to compensate for that?
Well, in my experience, I've never seen wet disinfection, you said, right?
Yes.
No, I never seen that at the transfer time. So, and either during the research of the studies, I don't see any research done on this side. So,
okay.
I don't know if Carolina has some feedback from her own experience, but.
Well, there are some hatcheries that they do it, but the best way to do it is in the hatchery, because that's the place where the hatching process is. So each time a chicken is hatching, it can get disinfected. That's the reason why it is better to go to the hatcher. Also, when you do it on transference, these wet eggs get inside the hatcher and it will increase humidity. So better to avoid that.
Okay. All right. Very good. Well, we do have again several other questions that seem to apply to other aspects of incubation, which we're going to answer offline here. They don't necessarily relate directly to eggshell quality and the factors that affect that. And so we'll certainly acknowledge every one of those questions privately and have a hatchery specialist return your call to talk about your issues. And so again, if anyone does need some more insight or would like to consult with us, so reach out to us at chickmaster.com. You'll find all of the ways to contact us, and we'd be happy to have that conversation with you. We are really looking forward to the time that we can again visit with you in person at your hatchery and to do everything we possibly can to help you get the best possible results. So with that, I'd like to say thank you to Carolina and to Mohamed and also to all of our guests. We have plenty of other webinars scheduled in the future on a variety of topics related to the whole incubation process, and I hope you'll keep taking advantage of those. You may get a survey right at the end of this call. Please give us some feedback on whether you found this useful or any suggestions you have, and we will be sending out a copy of the recording as well as some of the resources that Mohamed mentioned just within a couple days here. You'll get a follow-up so that you can review this yourself. So with that, thank you to all of our guests and to our speakers, and we will see you guys on the next webinar. Take care.